Hello, and welcome to Summarize Science. Have you ever wondered what the microscopic machines inside our cells actually look like? Scientists use a technique called cryo-electron microscopy, or Cryo-EM, to take thousands of 2D pictures of things like proteins. But turning those flat, noisy images into an accurate 3D model is a huge computational challenge. For years, there's been a trade-off. Traditional methods are fast, but they often lose fine details in the process. Newer, AI-based methods can create stunningly detailed models, but they're incredibly slow and require massive amounts of computer memory and power. This makes them impractical for many research labs. But this paper introduces a groundbreaking new method called GM. Instead of trying to model every single point in a 3D space, GM cleverly represents the protein using a collection of what you can think of as smart, 3D blobs called Gaussians. This is far more efficient because it only describes the parts where the protein actually exists, ignoring all the empty space. Each of these blobs is defined by just 11 values, making the entire model incredibly compact. And the results are staggering. As you can see in this table, GM is dramatically more efficient than its competitors. It trains up to 48 times faster and uses up to 12 times less memory. One of the competing methods even ran out of memory completely during testing. But here's the amazing part. It's not just faster, it's also better. These charts measure the final resolution, and for every single dataset, GM proves it achieves a higher quality reconstruction. Looking at the 3D models themselves, it's easy to see the difference. The blue and green areas represent higher resolution or finer detail. GEM's models are consistently more detailed than those from the leading methods. So let's recap the key points. The GM framework manages to be faster, more memory efficient, and more accurate than previous state-of-the-art methods. It breaks the long-standing trade-off between speed and quality in cryo-EM reconstruction. What does this mean for the future? This could dramatically accelerate research in areas like drug discovery and understanding diseases by making it faster and cheaper to determine the precise structure of biological molecules. It puts this incredibly powerful technology within reach of many more scientists around the world. This really is a game changer for structural biology. Thanks for joining us on Summarize Science. If you found this interesting, please like the video and subscribe for more scientific breakdowns.